Yeah. Thank you. So, hello everyone. I am Suvojit Hazra. I am working as a postdoctoral research associate under Professor Sushmita Roy at Wisconsin Institute for Discovery in the University of Wisconsin-Madison. I would like to thank ISMB organizer for giving me this wonderful opportunity to present our latest research work on phylogenetic dynamic regulatory module networks to study the evolutions of mammalian gene regulatory program. So we all know that cis-regulatory elements such as promoter and enhancer are abundant in the genome and drive the gene expression levels. Transcription factor bound to cis-regulatory elements constitute the backbone of gene regulatory networks. Changes in gene expression and its regulation significantly contribute to morphological diversity across species. Beyond sequences, comparative regulatory genomic studies collects and compare different regulatory signal from multiomics measurement. Comparative regulatory genomic studies offer a powerful framework to systematically study the role of gene regulation in the evolution of complex traits. Recently, certain studies have been published that incorporates the expression data and leverages the phylogenetic relationship across species to infer species-specific expression module like arboretum, regulatory networks like Martel, and subnetworks like Muscari. However, these studies lacks the integration of multiomic data. On the other hand, recently certain studies have been performed that integrates the multiomics measurement to infer the dynamic gene regulatory network in different contexts like developmental stages in certain time points or disease progression. Such example includes DRMN, DRAM, Chrome HMM, and omics integrator. However, computational tools that are phylogenetically aware and can measure such a high dimensional multiomics measurement for the study of evolutionary dynamic gene regulatory networks across species remains scarce. Therefore, we are introducing phylogenetic dynamic regulatory module networks that leverages the bulk multiomics data and use the phylogenetic relationship across species to study the dynamic regulatory module networks to study the evolution. So it is based on a novel multitask regression-based learning algorithm. Before going to P-DRMN demonstration, first I am going to tell you about what is a regulatory module network for one species. So in regulatory module network, we represent the gene expression with the module representation. A module is defined by a set of genes that having a particular expression state for a species, which we can interpret as a discrete expression level. On next, in regulatory module network, the component is regulatory program, which is composed of two parts. First is a graph structure, that means what are the regulator for genes, and second one is parameter, that indicates that how the regulator predicts the expression state. So as we are using the simple linear regression model, so the parameter would be regression coefficient. So the measured regulatory activity is the signal strength of the transcription factor and their upstream cis-regulatory element for a gene in a module. So we train the regression model in such a way that it predicts the expression based on the regulatory feature vector of those transcription factor and cis-regulatory elements from the multiomics measurement. So P-DRMN is a collection of multiple regulatory module networks that integrates the bulk multiomics data and leverages the phylogenetic relationship across species. And P-DRMN models the gene expression at the level of similarly expressing gene modules based on the low to high module assignment of the constituent gene, and it outputs the species-specific dynamic regulatory module network where a gene membership in a module is based on the regression function of its upstream cis-regulatory features. P-DRMN does this using a fused lasso objective where the phylogenetic relationship between species is learned during the regulatory programming learning. So uh, uh, now I am going to the applications of the P-DRMN on a data that was generated with the collaboration with the Professor Michael Wilson from Sick Hospital of U Toronto. So who is actively working on the endothelial cell systems. And endothelial cells, we know that it lines our blood vessels involved in vascular homeostasis, response to injury and inflammation, and it forms the blood vessels. And endothelial cell dysfunction is implicated in various devastating vascular diseases like atherosclerosis, irritable bowel disease, or cardiovascular disease. Wilson Lab have prepared the bulk multiomics measurement across different transcriptome and epigenome assays like RNA sequencing, attack sequencing, and chip sequencing of the 5 histone marks for the 5 mammalian species. For the aortic endothelial cells, the species are human, rat, cow, pig, and dog. So we are using this data to infer the evolutions of the gene regulatory program in aortic endothelial cells in mammals as an input for the P-DRMN modeling. So for P-DRMN, we prepared the feature by integrating the promoter accessibility and the motif feature set. In addition to this feature set, we have included the 5 histone marks feature to get the informations of the epigenetic regulation, as well as for the transcription factor binding motif. We are using the 2 different sets. One is the conserved regulator sets across 5 species we are studying, and also the cumulative all regulator set for this transcription factor. And we are collecting the gene centric feature information for the 3 different genomic regions. One is the TSS plus minus 2 kbp, plus minus 5 kbp, and 10 kbp. So a total of the 12 different feature combination were used as an input to P-DRMN model to get the most stable expression state learned from this program. So next we ask how well the P-DRMN predicts the expression. So by the study of the of the 12 different hyperparameter combination, we found that the all regulator features outperform the conserved regulatory features, and adding the histone mark significantly contribute to gene expression prediction. And we found that the best conformation that gives the most stable expression state is the when we consider the genomic window of the TSS plus minus 2 kbp for all regulator for the motif plus attack plus histone features. So now I'm going to demonstrate about the result of this configuration in the subsequent slides. So this is the overview of the how the module looks like from the P-DRMN learning. Here the blue color indicates the low expressing gene module, and the red color indicates the high expressing gene module. And the number represented here is the constituent gene present in each of the module. And we found that P-DRMN predicts the gene modules are quite conserved across species. On next, we calculated the module conservation across species for a module. Like that, we calculated the genes that are assigned to a module across species in a particular module, like human to rat, human to cow, human to pig, and human to dog, and so on, using the Jaccard index. And we found that the module conservation score ranging between 0.32 to 0.76. And we found that the high expressing gene module like module 3, module 4, and module 5 having the high conservation score, whereby the low expressing gene module like module 1 and module 2 having the low conservation score, means that they having the significant divergence across species. Next, we identified the biological function associated with those conserved gene module in the aortic endothelial cell of these 5 mammalian species. And we found that the low expressing gene modules are associated with the endothelial cell-specific biological functionalities, whereby the highest expressing gene module, that is the module 5, is associated with a function called angiogenesis. That means the new blood vessel development, which is the important functions of the aortic endothelial cell. We also observe that several human-specific pathways that are being activated in the highest expressing gene modules. All are these the immune signaling pathway like that NIK, NF, kappa, beta, WNT signaling, and MAPK signaling. So on next, on the P-DRMN inferred modules, we identified certain set of genes that changes the module assignment across species. These genes are known as the transitioning genes. As an example, you can see that the Hoxby gene is having the low module assignment in all studied species except for human. And in human, it changes the module assignment to a high expressing module state. So this is the classical example of the transitioning genes. And a set of transitioning genes that exhibit the same pattern across species and clades are known as the transitioning gene set. From the present study, we identified there are 11 transitioning gene set with diverse phylogenetic trends across species and clade. So now I'm going to demonstrate you about the two interesting transitioning gene set. One is the cowpea clade specific transitioning gene set, where you can see that in the cowpea clade there is a high module assignment, and this 10 genes changes to that. And on next, there is another transitioning gene set that is having the high gene expression module assignment in human rat compared to other species. Next, we try to identify the putative regulator for this transitioning gene set, and for that, we first consider the all genes in the transitioning gene set and consider the expression level for this transitioning gene set. Next, we incorporated the feature signals derived from the attack signals or the motive activity, which are learned from the P-DRMN inferred uh, genes present in a module. After that, we incorporated both the expression and the features into our mammalian linear regression based learning framework, and from there we identify the gene regulatory network, where the connections between the transcription factor of a gene is the regression weight. Next, we filter the network in such a way that we first calculated the correlation coefficient between the expression and the regulatory features of the corresponding regulators, and with a filtering of correlation coefficient of greater than equal to 0.6, we get the filtered gene regulatory network that having the strongest regulatory possibility. So by this way, from this two transitioning gene set, we identified MIXL1 and UNCX transcription factor is regulating this cowpea clade specific transitioning gene set, and on the other hand, a mouse specific transcription factor HSF1 and a histone mark H3K27SC is regulating the gene expression of this human rat clade specific transitioning gene set. So therefore, we found that a combination of transcription factor binding and histone marks contribute to changes in the gene expression across species. So taken together, P-DRMN offers a powerful framework to systematically study the role of gene regulation by incorporating the bulk multiomics data from different species by leveraging the phylogenetic relationship between them. So that's all for today. I would like to thank Royal Lab member, especially Sarah and Erika, and Wilson Lab member for their help in each and every possible way to complete this work. I'm also thankful to Wisconsin Institute for Discovery and University of Wisconsin-Madison and their high throughput computing facilities to carry out this work. I would also like to thank Department of Energy and James S. McDonnell Foundation for providing the necessary financial support. Thank you all. So please let me know if you have any question.
Hi, thanks very much for the talk. I wanted to ask how robust or complete does the data across the species need to be for this method?
Yes, actually, we found that the P-DRMN model predicts the expression at a range of around 80% across all species and the modules on an average. So we are thinking that it predicts the expression very well.
Thanks. Any other questions? All right, I'm going to take my prerogative then. So I was noticing on one of your plots, you were showing the performance of your ability to recover the expression modules as a function of your window size around the transcription start site.
Yes, yes, yes.
And it looked like the smaller windows seemed to outperform the larger window, and I think that was interesting, because I think sort of the lore out there is people look at 10 kb windows sort of as their default without really. And what do you think what do you think is the interpretation of finding that a smaller window size like 2 kb is outperforms the larger window size? Do you think it's because all of the signals actually in the smaller window, or do you think it could be that you just get more confounding signals as you increase your window size?
Yes, when we increase the window size, the possibility of predicting the expression state comes to lower, but not too much, not too much. So you can see that if I share this, so you can see this, all the correlations are not very much low, but I am thinking that the promoter signals are strong enough rather than the distal enhancers. So that is why that may be a possible reason that can explain the thing.
Thank you.
Yes.
All right, so if there are no more questions, let me first thank our speaker one more time.